The development of hybrid seed technologies has played a transformative role in the global agricultural sector, ensuring higher productivity, improved resistance to pests and diseases, and enhanced resilience to environmental stresses. The advent of synthetic biology has further revolutionized the field, enabling scientists to engineer crops with superior traits that were once considered unattainable through conventional breeding techniques. This documentary explores the evolution of hybrid seed technologies, emphasizing the role of synthetic biology in shaping the future of global food production. It delves into case studies, recent advancements, controversies, and the implications of synthetic biology in the sustainable development of agriculture. Through extensive evidence and real-world examples, this documentary provides a comprehensive analysis of how hybrid seed technologies, bolstered by synthetic biology, are driving the next agricultural revolution. Hybrid seed technologies have their roots in classical plant breeding, which began with early agricultural societies that selectively bred plants to enhance desirable traits. The first scientifically recognized hybrid crops emerged in the early 20th century with the work of George Harrison Shull, who demonstrated that inbred lines of maize could be crossed to produce superior hybrid offspring with higher yields. This discovery led to the development of hybrid maize in the United States, which significantly increased agricultural productivity during the Green Revolution of the mid-20th century. A hybrid seed technology relies on controlled crossbreeding between two genetically distinct parent lines to produce offspring with heterosis, or hybrid vigor, which results in improved growth rates, resilience, and yield potential. However, conventional hybrid breeding has limitations, including the time required for developing new hybrids, genetic bottlenecks, and the constraints imposed by naturally occurring genetic variations. With the emergence of synthetic biology, these limitations have been addressed by introducing precise genetic modifications and novel traits into hybrid crops, allowing for a more efficient and targeted approach to crop improvement. Synthetic biology is a multidisciplinary field that applies engineering principles to biology, enabling the design and construction of new biological systems. In agriculture, synthetic biology is used to modify the genetic composition of crops to enhance traits such as drought tolerance, pest resistance, and nutrient efficiency. Unlike traditional genetic modification techniques which rely on the transfer of existing genes between organisms, synthetic biology enables the creation of entirely new genetic circuits and metabolic pathways, allowing for the introduction of synthetic traits that do not exist in nature. One of the most significant contributions of synthetic biology to hybrid seed technology is the ability to precisely control gene expression, enabling scientists to develop hybrid crops that exhibit specific desirable characteristics. For example, synthetic biology has facilitated the development of crops with inducible sterility, which ensures that hybrid seeds cannot be saved and replanted, thereby maintaining their hybrid vigor across successive generations. This technology has been widely used in hybrid rice, maize, and canola, where hybrid seed production is critical for maintaining high yields. Additionally, synthetic biology has enabled the development of self-replicating hybrid seeds, which could potentially reduce farmers' dependence on purchasing new seeds each season. By designing genetic circuits that allow hybrids to retain their vigor for multiple generations, researchers are exploring ways to create more sustainable agricultural practices while maintaining the economic viability of seed companies. The integration of synthetic biology into hybrid seed development has led to groundbreaking advancements in crop improvement. Several case studies illustrate the transformative impact of these technologies. Case Study 1: Hybrid rice with enhanced drought tolerance. Rice is a staple crop for over half of the world's population, but its production is highly susceptible to water scarcity. Conventional breeding efforts to develop drought-tolerant rice varieties have had limited success due to the complex genetic interactions involved in drought resistance. However, researchers at the Chinese Academy of Agricultural Sciences have successfully developed hybrid rice varieties using synthetic biology tools that enhance the expression of drought-responsive genes. By introducing synthetic gene circuits that regulate water use efficiency, these hybrid rice varieties can maintain high yields even under prolonged drought conditions. Field trials conducted in arid regions of China and India have demonstrated a 25% increase in yield compared to conventional drought-tolerant varieties, providing a promising solution to food security challenges in water-scarce regions. Case Study 2. Insect-resistant maize for Sub-Saharan Africa. Maize is one of the most important cereal crops in Sub-Saharan Africa, but its production is severely affected by pests such as the fall armyworm. Conventional insect-resistant maize varieties rely on Bt, Bacillus thuringiensis genes, which have been effective but are increasingly facing resistance issues. Researchers at the African Agricultural Technology Foundation have utilized synthetic biology to develop hybrid maize varieties with multiple stacked insect resistance genes that function through different biochemical pathways. These hybrid seeds contain synthetic gene circuits that activate defense mechanisms only in response to pest attacks, reducing the risk of resistance development. Large-scale field trials in Kenya, Nigeria, and Tanzania have shown that these hybrid maize varieties reduce pest-related yield losses by over 40%, significantly improving food security for smallholder farmers. Case Study 3 are high-nutrient hybrid wheat in Europe. Wheat is a crucial crop for global food production, but conventional wheat varieties often lack essential micronutrients such as iron and zinc. A collaborative research initiative between the University of Cambridge and the European Union's Horizon 2020 program has developed high-nutrient hybrid wheat varieties using synthetic biology. By engineering synthetic metabolic pathways that enhance nutrient uptake and assimilation, scientists have created wheat varieties with 50% higher iron and zinc content compared to traditional hybrids. These biofortified wheat varieties have been introduced in several European countries, improving dietary nutrition without the need for additional fortification. Case Study 4. Hybrid canola with enhanced oil quality. Canola is a vital oilseed crop used for cooking oil and biofuel production. Conventional hybrid canola varieties have been optimized for yield, but their oil composition has remained largely unchanged. Researchers at the University of Saskatchewan have applied synthetic biology to develop hybrid canola varieties with modified lipid biosynthesis pathways, resulting in healthier oil profiles with higher levels of omega-3 fatty acids. These hybrid seeds have gained significant commercial interest, with several multinational agribusinesses investing in large-scale production. Despite its numerous advantages, the application of synthetic biology in hybrid seed development has raised several ethical, regulatory, and ecological concerns. One major issue is the potential for unintended genetic interactions that could affect non-target organisms or lead to unpredictable environmental consequences. For example, the introduction of synthetic gene circuits into hybrid crops may inadvertently alter soil microbiomes, affecting nutrient cycling and ecosystem stability. Another concern is the monopolization of hybrid seed technologies by large agribusiness corporations, which could limit access for small-scale farmers. The high costs associated with developing synthetic biology-based hybrid seeds often mean that these technologies remain out of reach for resource-poor farmers in developing countries. Efforts to address these concerns include the development of open-source synthetic biology platforms that allow public research institutions to participate in hybrid seed innovation. Additionally, the ethical implications of designing synthetic life forms in agriculture remain a topic of debate. While proponents argue that synthetic biology can enhance food security and reduce the environmental footprint of farming, critics warn against the potential for unforeseen consequences that may arise from the large-scale release of engineered crops. The future of hybrid seed technologies lies in the continued integration of synthetic biology with other emerging fields such as artificial intelligence, nanotechnology, and precision agriculture. Advances in gene editing technologies such as CRISPR-Cas9 have already accelerated the development of hybrid crops with improved traits, and further innovations in synthetic biology will enable even more precise modifications. One promising area of research is the development of hybrid seeds with self-regulating genetic circuits that allow crops to adapt dynamically to changing environmental conditions. By designing plants with synthetic sensors that detect and respond to stress factors such as heat, salinity, or pathogens, scientists aim to create resilient hybrid crops that require fewer chemical inputs while maintaining high yields. Moreover, synthetic biology has the potential to revolutionize hybrid seed production by enabling the development of synthetic Apomixis, a process that allows hybrid plants to reproduce clonally without losing their hybrid vigor. This breakthrough would eliminate the need for recurrent breeding, significantly reducing the cost and complexity of hybrid seed production. The development of hybrid seed technologies using synthetic biology represents a paradigm shift in modern agriculture. By enabling precise genetic modifications and novel trait enhancements, synthetic biology has unlocked new possibilities for crop improvement, addressing critical challenges such as food security, climate resilience, and nutritional enhancement. While ethical and regulatory concerns remain, ongoing research and innovation hold immense promise for the future of sustainable agriculture. As synthetic biology continues to evolve, its integration with hybrid seed technologies will play a pivotal role in shaping the global food landscape, ensuring a more productive and resilient agricultural system for future generations.